Many of us have known Tom for a long time, and particularly his work on very early kidney development. And so he's in the Department of Internal Medicine and Molecular Biology at UT Southwestern in the Nephrology Division. And he's going to tell us about interstitial cell heterogeneity and creating microenvironments for that regulate kidney development and maintenance. So Tom, I'm going to turn it over to you. And again, thank you very much for participating.
Okay, thank you. Everybody can see everything?
Yep.
Okay. So yeah, as Brad mentioned, I'm going to tell you kind of about my latest obsession that we've had for a couple of years here, and that is the role of stroma in kidney development and maintenance. And I don't probably need to tell anybody here, but I'll just remind everybody about the kidney. The kidney is composed of these distinct functional units known as the nephron. And different kidneys and vertebrates are really very similar in their nephrons, and the main difference is the number of nephrons that they actually have, with a mouse kidney having somewhere between 20 and 30,000 nephrons in the mature adult, whereas a human kidney will have somewhere between 500,000 and 1 million nephrons, give or take several million. And we know that nephron number is essential for optimal function. It's estimated that if you fall below kind of 10% of that average number, so in a human somewhere below 100,000 functioning nephrons, you're in end-stage renal disease. And besides nephron number, we also know that nephron pattern is essential for optimal function. So if we go up here to the schematic of a nephron, the nephron is patterned in this proximal tubule, the loop of Henle, the ascending and descending and ascending limbs, the distal tubule, and the collecting duct. And many of the defects that you see in genetic diseases of the kidney are actually defects in the maturation of the different segments of the kidney. So, as Brad mentioned, my lab has really been interested in kidney development, is interested in kidney development, and we always have been interested in kidney development. And we've really been interested for many years in the question of how you go from this early stage kidney, which this would be a section through a mouse at around 11.5 days after fertilization. And at this point, the nascent kidney consists of really 3 cell types and about 20,000 total cells. And then within just a couple of weeks, 3 weeks, you expand tremendously to a point where you have a mature kidney in the adult. It's estimated that there now have been identified to be over 30 different cell types in the adult, and with billions, perhaps trillions of cells in the mature organ. And so how do we go from this relatively non-complex structure to this incredibly complex structure? And we've known quite a bit about the cellular nature of kidney development for almost 100 years now. The kidney's always been a favorite of embryologists to study. And I'm going to just summarize it using this schematic from a recent review. And there are really 2 processes that direct kidney development, and one of them is known as branching morphogenesis. So in the early stages, when I showed you that image a moment ago, we have this epithelial structure known as the ureteric bud. And the ureteric bud over the embryonic period undergoes this bifurcations, dichotomous branching morphogenesis, and in some cases trifurcations, but mostly dichotomous branching morphogenesis, so that each ureteric bud tip becomes 2. So 1 becomes 2, 2 becomes 4, 4 becomes 8, etc. during the entire embryonic period. Now, sitting next to the ureteric bud tips is a population of what we call the metanephric mesenchyme. And there's actually 2 cell types within the metanephric mesenchyme. Here outlined in pink are what are known as the nephron progenitor cells. And the nephron progenitor cells actually undergo mesenchymal to epithelial transition to form an immature structure, epithelial structure known as the renal vesicle. The renal vesicle then undergoes morphogenesis, and all of the non-vascular portions of the nephron that I just outlined previously, so everything from the proximal tubule all the way through the distal tubule, also the Bowman's capsule, all come from this renal vesicle through morphogenesis. And the ureteric bud actually becomes the collecting duct system of the kidney. And at a point shortly after this S-shaped body forms, these two epithelia actually fuse together so that they have one continuous lumen, so that filtrate that is produced here at the level of the glomerulus actually gets passage along the length of the tubule into the collecting duct, and then eventually passed out as urine. And the reason I actually included this specific picture is that if you go and look at most reviews of kidney development, they focus on the ureteric bud tips and the nephron progenitors and giving rise to either branching or this MET event. But nobody ever really discusses this other cell type, which is kind of outlined here in gray, and that is the stroma of the kidney. We've known for quite some time that the stroma actually plays a critical role in kidney development. So this is, I believe, the first paper that ever showed that the stroma was involved. This came from Essing Lai's lab back in 1996. He showed that a gene that was only expressed in the kidney stroma, this gene called BF2, when they knocked it out, had severe defects in branching morphogenesis of the kidney, and also some defects in maturation of the nephrons. I'm just going to let you know that BF2 is now been renamed FOXD1, and I'll be talking about FOXD1 later on. So this was the first paper that actually suggested that the stroma actually had a role in normal kidney development. Although since this paper came out, there's been kind of a lot of controversy in terms of what FOXD1 or BF2 was actually doing, with some groups actually saying that the stroma really didn't have a normal role in kidney development. And in fact, when you knocked out FOXD1, what was happening is you were changing the molecular nature of the stroma, so you're getting a neomorphic phenotype within the stroma, and that was what was secondarily having these effects on kidney development. And I'm going to address that in a moment. But before I get into that, I just want to define to you guys what I mean, and well, I'm going to tell you what I mean when I talk about the stroma. And I think most people mean the same thing as me, but just to make sure we're all on the same page. And I've talked to you about the nephron, so the nephron epithelium, the collecting duct epithelia. We also know, marked here in red, that there's vasculature in the kidney, the endothelia, which is marked in red. The stroma, to me, is everything else that is not epithelial or endothelial. And it really consists of fibroblasts, mural cells, smooth muscle, and leukocytes. So what does the stroma do? Well, for years, people have really primarily thought of it as playing a role in structural support, so kind of being a scaffold for the nephrons and the vasculature so that they don't just kind of flop around and fall on top of each other. And this is probably through the fibroblasts producing, which I think they produce the vast majority of the extracellular matrix of the kidney, so the collagens and factors like that. There are also the stromas necessary for the contractile forces that you see in the kidney. So the pericytes, the vascular and epithelial smooth muscle, they all have contractility properties in which that are used to actually regulate blood pressure or to actually expel urine through the collecting duct system and out the ureter so that it gets passage into the bladder. The stroma is also has all of the endocrine function of the kidney, and that it produces EPO and renin. And this is really done by specialized mural cells within the kidney. Within the stroma, and this is something that a lot of people don't really think about, the stroma produces almost every type of growth factor ligand that we think about as being the major regulators of kidney development and growth. So Wnt's, BMPs, TGFs, retinoic acid, FGFs, VEGFs, they're all produced by different cell types, or they're all produced by the stroma, probably different cell types. And these most likely have a very important role in the growth and survival of the kidney. And then I add this on for Brad. I probably wouldn't have had this if I wasn't speaking here, but I did mention the leukocytes earlier. Included within the stroma, we do include all of this immunogenic role of the leukocytes. But for and I don't doubt that this plays a very important role, but for the purposes of my talk today, I'm not going to really be talking about the leukocytes, and that'll become clear why in just a moment. So I mentioned this gene FOXD1 before, and FOXD1, which BF2 is now called FOXD1. FOXD1 is expressed in a subset of the stroma that sits at the very cortex of the kidney. So this is an embryonic day 15.5 kidney that's been sectioned through. And in blue, we have a marker for the collecting duct, ureteric buds and collecting duct system. And surrounding the ureteric bud tips are the nephron progenitor cells that will give rise. So these are the stem cells that will give rise to the nephron itself. And then sitting cortical and surrounding the nephron progenitor cells is actually the FOXD1 labeled stroma. Now, several years ago, work from Annie McMahon's lab actually showed that this FOXD1 expressing cells were the progenitor cell type for all of the non-leukocyte populations of stroma in the kidney. So this is shown here. If you take a FOXD1 Cree mouse, so a mouse that expresses Cree or the regulatory elements of FOXD1, and you cross it to a Rosa reporter and then fate trace it, you see that all of the stromal cells outlined here in green have come from this small population of progenitor cells that were expressing FOXD1. And you can see the FOXD1 positive cells do not give rise to the epithelia, so either to the collecting ducts or to the nephron segments, which are dark here, basically. So my lab, like many others that studied kidney development, really focused on the interactions between the nephron progenitor population and the ureteric bud tips. But several years ago, we were trying to understand how the balance between nephron progenitor renewal and mesenchymal to epithelial transition was maintained. And we hypothesized that perhaps what was going on is that FOXD1 producing cells produced a signal that told the cells sitting on top to differentiate, and then the lack of signals coming from those cells were perhaps what was necessary for, to renew, and the lack of that signal was necessary for differentiation. So to test this hypothesis, we decided to cross the FOXD1 Cree mouse to mouse expressing the diphtheria toxin A subunit under the control of the Rosa 26 locus. So this is a lock-stop-locks DTA mouse. So now we drive diphtheria toxin A subunit in all of the FOXD1 derivatives, so all of the stroma. So we call these stromaless mutants, and that the stroma actually dies. And what we saw, the mice die shortly after birth. And what we saw is that when you look at an H&E of a wild type kidney, you can see the ureteric bud tips, and you can see the outline of the nephron progenitor cells here, and then all the stroma cells in between. And then you start to see these renal vesicles and S-shaped bodies forming down below. And what you actually got in the stromaless mutants was you got this enormous expansion of the nephron progenitor cells. The ureteric bud tips were still there, but you never saw this mesenchymal to epithelial transition. And I'm just going to outline that here with molecular markers. Here, looking at markers of nephron progenitor populations, so cited 1 and 6-2. You can see in the stromaless mutants, you get this enormous expansion of these nephron progenitor cells. And if we then go and look at a marker of the earliest signs of mesenchymal to epithelial transition, in this case it's LEF1, you can see it being expressed in these renal vesicles in a wild type kidney. You can see that LEF1 expressing cells expressing epithelia are never formed in the stromaless mutants. So this was kind of the opposite of what we thought the stroma was doing. And instead, the stroma seemed at this stage, it suggested that the stroma was playing a role in promoting the differentiation or this mesenchymal to epithelial transition of the kidney. Now, we would have loved to see if there were any other roles for the stroma, but because there were no nephrons actually in these kidneys, we couldn't do very much more with this. But we started to actually investigate the stroma in more detail to try to find out if it was possible that the stroma was playing multiple roles in differentiation of the nephrons. And the reason we thought that it was doing this is because a number of papers have since come out where people have knocked out genes either using FOXD1 Cree or genes that are primarily expressed in the stroma, and they found various phenotypes in the vasculature of the kidney, in the formation of the loops of Henle of the kidney. And so it suggested the stroma may actually play multiple roles in kidney development. But up until this point, people still were suggesting that the stroma was a relatively homogeneous population of cells, so that there was a progenitor population that expressed FOXD1, and then the rest of the stroma. And that these various factors that were expressed, that were necessary for development of the kidney when they were knocked out, that they suggested that it was not the stroma that was actually regulating the specificity and phenotypes that were arising in the knockouts. It was actually the responding cells within the nephron or within the endothelia itself that regulated the response to the stroma. So an example of this would be to say, oh, that the stroma expresses a ligand uniformly, but it's the nephron that expresses the receptor in a regionalized portion of the nephron, like the proximal tubule. And so then when you knock out a gene and the ligand in the stroma and get a defect in the proximal tubule, it was actually the pre-pattern of the proximal tubule that was regulating that process. But we thought it was possible that the stroma was actually a much more heterogeneous population than had previously been appreciated, and that we just hadn't had the tools to investigate this before. So with the advent of single-cell RNA sequencing, we have the capability to actually investigate how many cell types there are in the stroma. So we took E18.5 kidneys, and this was work done by a graduate student in my lab who has since moved on, Alicia England. And so Alicia once again took the FOXY1 reporter, FOXY1 Cree mice and crossed them to a Rosa 26 reporter, in this case Rosa 26 tomato. And then at E18.5, she dissociated the cells down to single cell level and then FACS sorted to isolate the stromal population. And then she did RNA-seq, single cell RNA-seq on those using the 10x genomics platform. And she sequenced about 10,000 individual stromal cells. And what her analysis showed, or actually the analysis done by a bioinformatician who works very closely with us, Chris Cheney, who's an assistant professor in internal medicine. What Chris's analysis actually showed was that there were 17 distinct clusters within the embryonic E18.5 mouse kidney. And that to us was a fairly shocking level of heterogeneity. I mean, we thought there would be more cell types than had previously been suggested, which was that previously people had said there were maybe 4 or 5 cell types, and we thought there might be more than that. 17 was surprising even to us. But as many of you know, with single cell RNA sequencing, you can just change the algorithms a little bit and you can get as many cell types as you want, or clusters, I should say, as you want. So we wanted to go through and actually validate that each of these clusters represented its own unique cell type. So we did this using in situ hybridization on E18.5 kidneys. And we started trying to assign identities to the different clusters that we had come out of our analysis. And so here's an example of the type of analysis we did. This is in situ hybridization with 2 different genes, AKR1B7 and DLK1, that came out as being enriched in some of our clusters. And you can see that AKR1B7 is actually expressed. I have outlined here the glomerulus, and you can see that AKR1B7 is expressed in the extraglomerular mesangial cells, and it's also expressed in the vascular smooth muscle. Whereas DLK1 is expressed specifically in the glomerular, in the mesangial cells of the glomerulus, so not the extraglomerular, but the glomerular mesangial cells, and then also in the smooth muscles surrounding the urothelium. And so when we start to do more and more genes, in situ of more and more genes, we can actually say that genes that cluster 16 actually represents the mesangium, and cluster 14 represents the smooth muscle. We cannot distinguish the extraglomerular mesangium from the smooth muscle. So, but we were able to go through and in this way actually determine that we had at least 12 different cell types in the kidney at E18.5, 12 different anatomically distinct cell types in the E18.5 kidney. And why don't we have 17? Well, there are several populations that we still don't have the resolution, and we're working on that using techniques like fluorescence in situ hybridization to actually distinguish, for instance, whether a proximal tubule fibroblast and a distal tubule fibroblast are distinct populations. We don't know the answer to that yet, but we think that we will probably at some point show that there's at least 17 populations and perhaps more, because for example, these 2 populations, juxtaglomerular apparatus and the vascular smooth muscle, are still in the same cluster, yet we know they're quite different cell types. And so we think we're a little bit under clustered at this point. But at least at this point, we know that there are 12 anatomically distinct populations of stroma. So what else did we find besides some of these known ones that we expected to find? Well, kind of the most surprising finding that we actually got out of this was that there were stromal cell types that extended from the cortex down into the papilla of the kidney. Kind of forming this, what we call rings on a tree type of cluster, type of pattern. So clusters 1 through 3 being out at the very cortex. 1 through 3 are all FOXD1 expressing, but we can actually find that there are at least 3 different populations within the FOXD1 expression domain. And we have evidence that they may give rise to distinct subsets of stroma. So this progenitor population is not quite multipotent. It may be that there are somewhat less potent or pluripotent cell types within the FOXD1 population itself. And then there are these other cell types that are more medullary to the FOXY1 population. And when we started to look more closely at their expression patterns, we found that these different clusters actually mapped very nicely to the different portions of the developing kidney and the E18.5 pattern to nephron. So as I mentioned, 1 through 3 being the progenitors, 4 and 5 are actually what we're calling a transit amplifying population. So we think these are the proliferating cells of the stroma. 6 through 8 are cells that sit around the proximal tubule. I'll talk more about these in a moment. 9 sits just underneath the proximal tubules. 10 and 11 kind of sit around the thick portions of the loop of Henle. 12 around the thin portions. Sorry, and 13 down around the ureter itself. And like I said, we think it's possible that, for instance, this population 6 through 8, we know that there are fibroblast populations in there, pericyte populations in there, and we think it's possible that there are multiple different fibroblasts. So for example, we think the proximal tubule may have its own fibroblast, and the distal tubule may have its own fibroblasts that are molecularly distinct, but we just don't have the resolution. We don't have the antibodies to test that. And right now we're trying to generate tools to test some of these hypotheses. So one of the things that one might think is that the nephron patterning might reflect the stromal patterning. So is the stroma actually directing the maturation of the different segments of the kidney tubule? And I just want to, somebody may ask, what about the opposite? Maybe the nephron patterning is actually patterning the stroma. We ruled that out by actually looking at the patterning in a mutant that did not have nephrons, a WNT9B mutant. And we actually can find, when we look at that most of the stromal patterning actually exists even if you don't have a nephron. So the stromal patterning comes before the nephron patterning does, and it does not require nephron patterning. So what about the opposite? Well, how do we test this? The way we'd like to be able to do it is that we could go in and specifically manipulate at a genetic level every one of these cell types with its own specific Cree line, and that is something we are actually trying to do. But we don't have the tools to do that right now. We're trying to make other tools to start to really ask these questions in a really fine, controlled manner. We're not quite there yet, but I think we will be in about a year or two. But in the meantime, we've been taking a different approach, and that is to go back and take advantage of our single-cell RNA-seq data. And you can take expression of the genes in each of these different clusters, and you can group them according to pathway activation. So in this case, we've done what's called a Regulon analysis, where we have taken transcription factors and we have asked what transcription factors, for all of the transcription factors that we have kind of consensus binding sites and target sites for them, we've asked which of those transcription factors are actually active in the different clusters of the stroma and the embryonic kidney. And that's just mapped out here, where we have the different clusters along this top axis here. Each one of these black or white lines actually represents a cell. So you can see kind of the different numbers of the different stromal clusters just by how broad these lines are. And a black line means that the transcription factor is actually active in that cluster, and white line means that it's not active. And so if you just kind of blur your eyes a little bit and stare at this entire pattern, you can see that each cluster has its own distinct pattern of transcription factors that are active in it. And we can now then go and say, what happens if we manipulate those specific transcription factors that are only expressed or only active in a specific stromal cell type? What kinds of phenotypes do we get? What is the result on kidney development? And there's a lot of transcription factors here. There's a lot of different cell types here. We can't possibly look at all of them at the same time. So we tried to be somewhat focused, and we're not completely focused, but we're trying to focus a little bit. And at least part of our focus has been on this population, these clusters 6, 7, 8, and 9, which, as I told you a moment ago, actually represents the stroma that is surrounding the proximal and distal tubules, and then also this subproximal tubule population. And the reason we focused on these is because these were populations of cells that had never actually been shown before. Nobody had ever identified these unique cell types, and we wanted to actually see what they were doing in the development of the nephron epithelium. So what do these different cell types represent? these clusters, I should say, represent? Well, we think they represent at least 3 different cell types. So this is take an analysis where we look at the genes that are enriched in each one of these clusters, and then go and look at it, do a literature search basically, and say what terms or what cell types are associated with the genes that come up as being enriched here. And based on this analysis, we actually believe, and there's more than just the literature analysis, but the population 6 is a, I don't know, I don't have the, yeah, I'm sorry, somehow lost the legend here, unless it's over to the side and I can't see it. Oh no, you guys can see it, I can't see it because I have this thing here. Let me just move this. Population 6 represents a localized pericyte population. It expresses multiple different canonical markers of pericytes or capillary associated cells. 7 and 8 are fibroblast populations. Once again, they are regionalized fibroblast populations. We don't know if one of these is for the proximal tubule and one for the distal tubule. That's something we're still investigating. And then population 9 is actually expresses multiple consensus markers of a myofibroblast. And I'll show you in a moment, there's really no disputing that population 9 is a myofibroblast cell type. And kind of an important just side point here is that most people think of a myofibroblast as being a cell type that only exists in response to injury. But in the embryonic kidney, the myofibroblast, it seems to be a completely normal cell type. And we don't know if it represents some sort of progenitor-like cell or some sort of plastic cell that may be able to give rise to both smooth muscle and fibroblasts. And that's something we're quite interested in. Okay, but so, what's going on? So if we go and we look at our Regulon analysis and we act, what kind of transcription factors are active in cluster 6 to 9, we actually found that these 2 transcription factors, these are orthologs of each other, YAP and TAZ, are actually active in this region here, which represents 6, 7, 8, and 9. And just to remind you what YAP and TAZ are, YAP and TAZ are part of this highly conserved signaling nexus known as the Hippo-Warts pathway. They are regulated by their subcellular localization so that the phosphorylated form of the protein, so phospho-YAP, is actually out in the cytoplasm after it's phosphorylated by its upstream kinase called LATs or warts. If it's not phosphorylated, it moves into the nucleus where it drives transcription. And there it can drive genes involved in proliferation, differentiation, and regeneration. So I just want to show you, if we look at an antibody for YAP, a total YAP, you can see that YAP is broadly expressed in the stroma of the kidney. If you look out here in the cortex, you can see it's kind of diffuse staining, but as you move down into the medulla, around the proximal tubules and into that subproximal tubular domain, you can see it's primarily nuclear. If we look at phosphorylated YAP, which remember is the inactive YAP, it is still present, but it's only out in the cortex, and you can see it's predominantly cytoplasmic. So this supports the idea that YAP is active in the proximal tubule stroma. If we knock out YAP specifically in the stroma using FOXY1-CRE, I was very excited when we first saw these mice because I do have an interest in polycystic kidney disease. And when you were looking at the whole kidneys of these, you could see these big holes in the kidney. I thought they were cysts, but then when we sectioned through them, they didn't quite look like cysts. In fact, some people said, oh, they just look like you don't know how to section and you're just torn your tissue. But we had Marty Cohen's lab at the University of Florida did some nano CT for us. And they showed that there are just these huge cavities that are forming within the kidney. Now, why don't we think they're cysts? Well, if we go and look to see if they're lined by any sort of epithelia, they are not. We've looked at different markers for all the epithelial segments. The cysts are never, or the spaces are never lined by epithelia. We also looked at vasculature, thinking maybe there was some sort of hemorrhage. They're not lined by endothelial cells either. And in fact, they're kind of reminiscent of some structures that Sue Quaggan's lab showed several years ago, where when she knocked out the angiopoietin 1 and 2. And what Sue found in these structures was that they also did not have epithelial or endothelial cells lining them. But instead, she said they were lined by smooth muscle. And she said these were smooth muscle cysts. And because smooth muscle comes from the FOXY1 population, we thought maybe these were smooth muscle cysts. But we went and looked at markers of smooth muscle to see if they lined our cysts. And not only did we see, or myofibroblasts, not only did we see that they were not lining the cysts, we actually saw significant decrease in the number of myofibroblasts or smooth muscle cells, and really just myofibroblast cells in our mutants. Now, to ask whether these cells were actually lined by perhaps an undifferentiated stromal cell, we did lineage tracing, where we crossed in the rows of reporter, and then took a look, and we saw that this is one of these holes here. You can see, although there are stromal cells marked in green here, there are also endothelial cells and other what appear to be epithelial cells. So these structures are really not lined by anything, which has led us to conclude that they're actually just interstitial edema. So it's just fluid-filled spaces. And so we think fluid is being pumped into the interstitial space of the kidney, and it's just kind of crushing from within. So we did RNA-seq to figure out what was going on here. And just, I want to show you that when we look at the RNA-seq and say, and compare it to our single-cell RNA-seq data and say, what cell types are affected in our mutants, the only cell types that are negatively affected in our mutants are 6, 7, 8, and 9. So the cell types that were YAP and TAZ are enriched. So this really does seem to be a specific effect on the development of 6, 7, 8, and 9. And this is just to kind of show you this is the case. If we look at markers of the different populations, so lysyl oxidase, which is expressed in 6 through 9, is missing in our mutants. SMOC2, which is only expressed in 9, missing in our mutants. But if you looked at markers of other cell types, such as ABCDD1, which is expressed down in 11 and 12, these more medullary cell types, it's present, and in fact, it looks to be expanded in our mutants cortically. So what else does the RNA-seq tell us? When we did RNA-seq and looked for genes that were downregulated, because YAP and TAZ are thought to be primarily activators, we saw extracellular matrix organization defects, as we kind of would have expected, since the stroma produces the ECM. But one of the things that was surprising to us is that two of the most enriched pathways were vascular development and blood vessel development. And this was surprising to us because our preliminary analysis, we kind of ruled out defects of the vasculature because we saw all these endothelial cells and we didn't really see a defect in the formation of the endothelium. However, the genes that we had been looking at, PKM and endomucin, were generalized markers of the endothelium, but the endothelium, like the stroma, is a very heterogeneous population of cells in the kidney. So you have capillaries and large blood vessels, arterioles and arteries, vasa recta, etc. And when you look at a plastic cast of the kidney, you can just look at the vasculature, you can see that it's patterned along this cortical medullary axis in a pattern that is really reminiscent of the stromal patterning that we saw before, or different domains or clusters. So we thought maybe what's going on here, so we're getting defects in the patterning of the vasculature. So if we took a look at some of the genes that were downregulated that are specific markers of various endothelial cell types, such as VEGF receptor 2, VEGF receptor 2 is normally expressed only in the peritubular capillaries, whereas endomucin is a general endothelial marker. And if we looked in the YAP/TAZ mutants, although they still had blood vessels, they didn't express VEGF receptor 2. That's just highlighted here. And we asked the question of, is there anything specific, anything special about these peritubular capillaries? And one of the things we know about the peritubular capillaries is they are not a normal endothelium. So a normal endothelium that we think of as a cells that are connected by cell-cell junctions, it's fluid tight. The peritubular capillaries actually form a fenestrated capillary where they have these little holes in them that allow fluids to pass freely from one side of the capillary to the other side. And one of the specific markers of a fenestrated endothelium is PLVAP. It actually is expressed in the membrane that sits on top of these fenestrations. And you can see here in green that it is expressed in the peritubular capillaries of a wild-type kidney, but in the FOXY1-3 YAP/TAZ mutants, a PLVAP is not expressed in the capillaries, and that's just shown highlighted here. So we think that this may be explaining the defect that we see in the YAP/TAZ mutants. So in a normal proximal tubule, you have filtrate that gets passages along through the interstitium, gets put back into the fenestrated capillaries, passing through these fenestrations, and then get put back into circulation. And in a YAP/TAZ mutant, because there are no fenestrations, that filtrate cannot get into the capillaries. It cannot get put back into the circulation. It just builds up in the interstitial space, creating these large lumens that large structures that we see that are these fluid-filled holes, basically, edema. So does this explain the phenotype completely? Well, maybe, except somebody pointed out to us, many people have asked us, what about the proximal tubules? Are they mature? And we didn't really have a great way to look at that until this paper from Joseph Park came out in 2020. And Joseph actually showed that in the E18.5 kidney, there were 3 stages of proximal tubule maturation. There was a very early proximal tubule that was marked only by cadherin 6. There was a later proximal tubule that had cadherin 6 and low levels of LTL. And then there was a more differentiated proximal tubule that only has LTL and very little cadherin 6. And when we stained our mutants with these 2 markers, cadherin 6 and LTL, what we found is we got the early structures, cadherin 6 positive only, and then the intermediate structures that are cadherin 6 and LTL positive, but we never got these LTL high, cadherin 6 low structures. So we definitely see a defect in maturation of the proximal tubules. Now, whether this is because we've lost one of the fibroblast populations, or it's an indirect effect of defects in peritubular capillaries, we don't actually know. That's something we're looking into. So YAP and TAZ, I've told you, are active in populations 6 through 9, which represent these regional fibroblast, pericytes, and myofibroblasts. And YAP and TAZ are necessary for the specification and differentiation of all 3 of those different stromal cell types. And then they have these indirect effects on the adjacent epithelium and endothelium. So we next ask whether YAP and TAZ are actually necessary for, or where does the specificity for YAP and TAZ come from? And I'm going to whiz through this real quickly because I see that I'm pretty much out of time. But we found in one of the populations, population 9, that myofibroblast population, that all of the YAP/TAZ target genes actually had consensus, or most of them had consensus sites for SRF right next to them. And SRF is known as serum response factor. It's very similar to YAP and TAZ. It is a factor that interacts with a cofactor known as MRTF. And SRF freely passes into the nucleus, but MRTF itself is regulated in its nuclear localization, and it is regulated by the actin cytoskeleton, so that if you have G-actin or globular actin, MRTF is actually repressed and maintained in the cytoplasm. But if you have filamentous actin, MRTF can go into the nucleus and interact with turn on target genes. Now, YAP and TAZ have before been shown that they regulate a number of the same genes, especially genes in myofibroblast differentiation. And there's been this question as to whether they act independent or they cooperate with each other or both. And this is some work from Tijan's lab, where he actually suggests that MRTF and SRF have independent roles and shared roles turning on different subsets of genes. So we wondered if YAP might be interacting with SRF in the stroma, and that's how YAP was at least specifying that myofibroblast population. So we knocked out SRF in the stroma. We got a phenotype that was somewhat reminiscent of the YAP/TAZ mutants and got very loose fibroblasts, but we did not see this interstitial edema that we see in the YAP/TAZ mutants. If we then went and characterized these mutants, we actually see that unlike YAP/TAZ mutants, the population 6 through 8 form completely normally. We get normal development of the peritubular capillaries. So all of the different cell types that form normally that are absent in the YAP/TAZ mutant, or most of them are normal in an SRF mutant. The cell types that form normally in a YAP/TAZ mutant are normal in an SRF mutant. The only cell type that is affected about knockout of SRF is this myofibroblast population. And we actually showed by activating YAP at the same time that we knocked out SRF, that SRF and YAP/TAZ are actually cooperate to specify this myofibroblast population. So in summary, what our data actually suggests is that YAP and TAZ and MRTF, SRF have unique targets, but they also have a shared population within the kidney that specifies the myofibroblast cell type. And I'm going to skip over this, although it's probably the only thing that many of you are interested in. And I'll just really quickly just say that similar level of interstitial heterogeneity exists in the adult. And we think that in the adult, it's necessary for maintaining the different cell types in the adult epithelia, and we have some data that actually supports that. And I'll just remind people, you're probably, all of you are very aware of Brad's work with the macrophages and other leukocyte populations that are necessary, playing a role in polycystic kidney disease. This is work that we had with Vishal Patel here, where we knocked out this interstitial this microRNA miR214 specifically in the FOXY1 population, show that it attenuates polycystic kidney disease. So I think that the populations in the adult interstitium, the heterogeneity that exists there, is probably going to play an important role, not just in maintenance of the epithelium, but also in disease states. So I will just very quickly summarize here. Hope I convince you that the stroma is heterogeneous in at least 12 anatomically distinct populations. Stromal signals act non-autonomously and are necessary for multiple steps of kidney epithelium and endothelial formation. The proximal tubule stroma is necessary for growth and maturation of proximal tubules and differentiation of peritubular capillaries. Stromal heterogeneity exists in the adult and the renal stroma. Finally, the big point is the renal stroma creates unique niches that are crucial for renal differentiation and function. And I tried to mention the people that did this, and so I'll stop there and take questions. And like I said, I'm sorry I ran long.
Tom, that was wonderful. Thank you. This is now open for questions. I'll actually start off. You know, in PKD, a lot of times the cystic epithelium is de-differentiated. Same thing kind of like that happens with after injury. Do you know, does that impact the differentiation state of the stroma around those cells?
So I almost put this in there and I took it out at the end just because I thought I was going to run long, and I did anyway. One of the first ways we got involved in this, Brad, looking at the stroma, was that some of the genes that we were looking at when we were looking at the various PKD mutants, when we did RNA-seq on some of the PKD mutants that we were looking at before, we saw a bunch of our stromal genes were downregulated. And so when we look at the markers of the pattern stroma in the adult, many of them are lost in PKD. So we think there's probably something going on with the crosstalk. So if you de-differentiate your epithelia, you're losing some sort of crosstalk with the stroma that sits next to it, and then you lose the differentiation of the stroma itself. So in the adult kidney, I have no doubt that there's crosstalk between these things that's maintaining, or I think that it's maintaining the identity of both. And I believe that that actually will contribute in some way to polycystic kidney disease. And you and I have talked about this, but whether it's doing that by directly signaling to the epithelia or the endothelia, you know, which Terry might think about, but also, or whether it's just signaling or interacting with the macrophages and other immune populations in there. That's what's the direct driver. I don't know. But I think the answer to the question is that I think that we know for sure. I have multiple examples of defects in stromal patterning and polycystic kidney disease.
Cool. And other questions? I don't know if there's anything in the chat or.
We still have a minute.
So I'm going to ask a very selfish question. Oh, go ahead, Sasha.
Yeah. Hi, Tom. Thanks for the great talk. I was just wondering if you look at the lymphatic system in, I forgot what the mutant is, the one with large cavity.
We looked at both. So we, strangely in the YAPTAZ mutants, everybody kind of asked us that same question, whether we see no defect in the lymphatics. So, you know, whether they're functioning or not, at least all the markers we looked at, they're patterned correctly, they're present, they're not, they don't seem to be a defect. In the SRF mutants, the lymphatics are basically almost completely gone. So, and you know, that's Sue Quaggan attributed those big spaces, interstitial spaces she sees in her mutants, defects in the lymphatics. And so we looked at that thinking that we would see the same thing. We don't. And so we think the lymphatic defects are probably contributing to kind of that loose mesenchyme or loose stroma that we see in the SRF mutants. And, you know, one of the things I want to point out on that is that, you know, some of the things that she saw in the lymphatics and attributed to the lymphatics, you know, these peritubular capillaries, especially the expression of PLVAP, I think that right now, you know, the patterning of the endothelium of the kidney is not very well understood. And I think that, you know, maybe some of the things that Sue was seeing could also be contributed to defects in patterning of the endothelium and not of the vascular endothelium, not the lymphatic endothelium.
Get back to my selfish question. Are there cilia present on the interstitial cells? I have never looked, to be honest.
You know, I can't, I have looked. I can't remember the answer to that. Okay. I know that we did knock out KIF26 in there to see if we got a big defect long ago. And once again, I don't think it had a very pronounced or profound phenotype because we never followed up on it, although I don't know the answer to that. We thought about it.
I did see that one of your transcription factors was GLE1.
Yep.
Which would suggest that there has to be some component of a cilia there somewhere for a response.
I mean, it's possible, GLE1 is also present in Drosophila, and they don't have cilia, right? So, I mean, it's possible there are some cell types that don't need cilia for hedgehog regulation. So, yeah, but GLE1 is one of the major regulators of the stromal patterning also on hedgehog cilia.
Other questions?
May I ask a question? So you performed the single cell sequencing for the proximal tubule stroma. So we know that the single cell analysis, they have a trajectory analysis, then can direct the differentiation or the how to say how the cell type will be transient to another cell type. For example, for the proximal tubule cell, whether they can transit to the fibroblast or the microfibroblast, and in which direction they have the trajectory analysis. I'm not sure whether your paper show that or not.
Yeah, we did do a trajectory analysis in the paper, but I'm just going to tell you. Well, sorry. We kind of thought that what we would see was that, if you look at these cell types, and I don't know why we thought this, but we thought, can you see this still?
Yes.
We kind of assumed that because FOXY1 was out here in the cortex, and then we got all this differentiated down the medulla, that we would see that FOXY1 gave 1 through 3, gave rise to 4, 5, gave rise to 6, 8, gave rise to 9, and then we would see this lineage trajectory analysis. We don't see this, and we've now actually the trajectory analysis that we did with our single cell actually suggests that the cortical interstitium gives rise to population 10, which is this population of cells that sits here. And then those cells then go this way and give rise to 11 through 12. I was supposed to say 11 through 12, not 10 through 12. And then also go up and give rise to the more proximal tubule. And that the last population that actually forms is this proximal tubule interstitium. And we have some lineage analysis with Cree lines that actually suggests that that is indeed the case. And so right now what we're trying to do is actually see what happens in the adult, and do these populations match different populations in the adult. And so we've been doing, we now have single cell for multiple time points in the embryo and in the adult going out to 12 weeks, and actually trying to do a much more detailed trajectory analysis, and then also do some lineage tracing. But so far, it actually suggested it is not this kind of linear development as suggested, but instead this more medullary population is the second to arise, and this may actually function as another progenitor population to give rise to everything else. I think that's an incredibly interesting question, and something that we are actively pursuing.
Yeah, because that program also can tell you the decisive genes. Did you find the SRF in that in the list of decisive genes for the transition between the cell types?
You know what, I don't know actually if, and I know what you're saying, and I don't know if we actually did. I mean, the way we pulled out SRF was that we were just looking at the genes, the YAPTAZ target genes through, the bulk RNA-seq, and looking for conserved sequences. But whether it is actually one of the genes that is kind of one of these hallmarks of these different anchor populations, I don't know. It's something we could go back and look at. But that is the kind of stuff that we're doing, is actually looking at the genes that look to be involved in progressing from one state to another. And that is another way that we're kind of trying to direct our future studies to study the role of these different populations. It's a really good question. Good point.
Okay, thank you.
Questions? Tom, I have one more then if nobody else does. So I know you do a lot of organoid stuff, right? If it's possible to replace during the differentiation stroma from different parts, does it change the fate of those cells? I know you're doing something.
Or of the epithelia.
Either.
So you kind of know this, and you may not know that you know this, but So I think with Brad, one of the things we started trying to do in the earliest stages of this was we made stromal organoids, and we have, and I didn't show this, we have reporters for all of these, or many of these stromal populations now. And so we would make an organoid where we would go and purify and just get the nephron epithelial progenitors and set them aside, get rid of all the stroma, and then fact sort and collect specific stromal populations and put them back together. And we'd already done some lineage tracing, which suggested that these cell types, the stromal cell types, most of them were stable, so that they weren't, as, which I just, with Xiaogang's question there, that they weren't just one giving rise to the other. And so they looked like they were somewhat stable, so we thought we were in great shape. Every single stromal cell type, when we put it back into the context of an organoid, it basically ended up dedifferentiating, becoming a progenitor, a multi-protein progenitor, and giving rise to every different stromal cell type in an organoid. So we said they were stable, and we now call them metastable. Under normal conditions, they're stable. They maintain their phenotype, their molecular phenotype. But once you disrupt them and put them in this, screw up the kind of the niche signaling, they revert to this progenitor population, which I think is actually super interesting when you think about injury. Maybe the same thing happens in injury. If you injure the epithelia, now these cells don't have this crosstalk anymore. Maybe they can now dedifferentiate and then turn into different stromal cell types to try to replace that. So the way we're trying to get around this is working with Leif Oxberg, who has gone through and tried to make stable cell lines for each of the different cell types. And now we're going back and doing that. And at least with the proximal tubule stromal cell type, when we put that with nephron progenitors, we can convert almost all of the cells to form proximal tubule. And we get very little distal tubular loop of Henle in those. So I wish we could do that with a primary cell type, but we can't. But at least with that immortalized cell type, then it's conditionally immortalized, so it doesn't just overgrow heat shock inducible. That actually can push these cells towards becoming a proximal tubule. That's the only one that we have tested so far. I'll say we've actually tested one that's a proliferative cell type for those transit amplifying cells, and it now just makes the nephrons proliferate like crazy. And that's something I would love to look to see if that cell type is overrepresented in polycystic kidney disease and maybe facilitate proliferation.
One more comment, because they have now they develop a program called cell chat. They can understand they can predict the cell-to-cell communication by analyze the single cell sequencing data. So maybe because you mentioned about the interstitial cell, proximal tubule cell, all the others, maybe you can reanalyze your single cell sequence data again, because that program, I'm not sure whether when you perform the single cell sequence analysis, whether that program available or not, but I now know it's available. So we really want to know the cell-cell communication in the cystic kidneys in the PKD.
Yeah, cell chat, is that what you said it's called?
Yes, cell chat program.
So I don't know if our guy that does bioinformatics knows about that. We've done kind of receptor ligand analysis, and we've also doing spatial transcriptomics so that we can actually see which cells are associated with the others and then do more informed receptor ligand and also looking at metabolite signaling. But if this is something that's more sophisticated, then yes, I would love to take a look at that, because that's really the direction we would like to go. Yeah.
Thank you. Yeah.
We are out of time. Tom, I want to thank you again for a wonderful seminar. If there's anything that the center can do to help with your research, please don't hesitate to reach out to us. We have a lot of resources up there, biobank of materials and from human and so forth, if you ever need any of that. So
thank you very much. Thanks for your questions. Thanks for having me.
Yeah, thank you guys. Appreciate it.
Yep. Bye.